In this video, we're going to talk about the next step in vision. So we've gotten light into the eye through the cornea and the pupil and the lens back to the retina. We've projected it, we've accommodated for light to project it to the right place on the retina. Next step is to talk about what goes on at the retina as we receive that signal, and then where the signal goes from there after it's transduced and turned into action potentials. Eventually, we're going to get to the occipital lobe, which you know is in the posterior part of your brain where that signal is going to be processed. So here's a zoom in on the retina. So there's an interesting thing that happens in the retina where the light is heading to the back of the retina, to the back of the eyeball, and then the processing actually goes in reverse. So the light's going to kind of pass through a few layers of cells, and then it's going to get processed right at the back of the eyeball where the light projects to, and then the processing is going to move in this opposite direction, and then eventually be sent back to the brain via something called the optic nerve. What you see on this image is primary receptors called rods and cones. These are going to receive the signal first. The signal's then going to pass through these bipolar cells. They're bipolar because they have one pole here at the receptors and another pole here at what are called retinal ganglion cells. Retinal ganglion cells are neurons. Okay, so this is going to be the first part of what we might consider your nervous system. So you got receptors here, connect to bipolar cells, which connect to retinal ganglion cells, which are neurons. Inside, we've got a couple of different types of cells called horizontal cells that move horizontally, and then amacrine cells that also move horizontally, but they got a different name. What these are going to do is going to coordinate activity across the receptors. So this one's connecting this and this and this, or across the ganglion cells, this, right? So we don't just have a signal going back and forth one to one. We've got some coordination between them, and this is going to help us to see in more complex ways. We'll talk about that a bit more in the next video. Well, this is a general idea, right? We've got receptors here, rods and cones, to bipolar cells, to retinal ganglion cells. And then in the process, we've got these other two cells, horizontal and amacrine, that are going to coordinate some activity. Cool. So all this is going on in the back of your eye. There's a few features of that retina that are really important to how we see. And the first feature is that the center of your retina is a bit different than the rest of your eye. And the center of your retina is referred to as the fovea. Sometimes it's also referred to as the macula, but the fovea is the term we're going to use here. And if we're taking in light and we're moving our eyes to take in light directly, like if you want to read something, you look directly at it, and then the lens is accommodating and adjusting the light to get to the back of the eye to a certain place, then there must be something important about the center, right? So the very center of your retina is very important for seeing clearly, right? So that's where we focus. Well, what's going on in that fovea? Well, there's a couple of features. First, you can see here in the fovea that there's fewer of these non-receptor layers. So you got the receptors here, and then there's fewer or smaller layers of the other areas. So we're concentrating on receptors, rods and cones. But in fact, not rods. All of the receptors here in the fovea, or basically all, are cones and not rods. Okay, so the fovea is different. It's focused on receiving, and it's focused on cones. We'll talk in a minute about what's different about the rods and cones, but just so you know that this is what this is going to do, focusing on receiving and focusing on cones, is going to lead to high acuity vision. By high acuity, I just mean clarity. So the center of your eye is clearer, and the reason for that, which we'll explain in a minute, is that we have a high density of cones. A couple other features of the retina include the optic nerve, which is going to come out the back. That optic nerve that comes out of the back of your eye and then is going to project back to your occipital lobe, eventually connect to a couple other things, including the thalamus. That optic nerve is this band of neurons come from the retinal ganglion cells. There's a place in the back of your eye where this optic nerve needs to connect, right? So the optic nerve needs to come in from the back and needs to have its retinal ganglion cells projected onto the retina here. There's this whole spot here called the optic disc. This is where the optic nerve connects to the eye. Interesting phenomenon is that because the nerve is connecting here, if the light comes in, it's going to hit blood vessels and retinal ganglion cells. It's not going to hit any receptors, right? No rods, no cones. So this is technically a blind spot in our visual field. Any light that comes here is not going to be seen because there aren't any receptors to receive it. Now your book has a nice demonstration of this. You can't really demonstrate it on video, but there's a certain spot where if you close one eye, so you're only getting signals to one blind spot, and you move a certain distance, that you can see where objects in your blind spot will actually disappear. Okay, so we're walking around with blind spots in our eyes all the time. It's an interesting thing because we don't experience that, right? We don't perceive it usually. We're filling in the blanks with our minds. Okay, so the signal comes in, it's got a blind spot. We're going to fill in that gap with everything that surrounds it and make assumptions about what's in there. We also have two different eyes, right? So whereas one eye might not be able to see this spot, the other eye might be able to. And so we can fill in the gaps because we've got multiple signals. Okay. So this is all going on the retina. The retina's got that fovea in the center, and then actually pretty close to it, we have a blind spot caused by the optic disc where the optic nerve connects to the back. I promise you we come back to the rods and the cones and how the cones especially are going to help us to see clearly. You can see pictures of the rods and the cones here. They get their names just because of what they look like. So these are rods that look like rods, and these are cones that are a little more cone-shaped. That's all the name means. There isn't anything else about the function of them or what they're called. However, they do have different functions. So cones and rods work differently. And the theory for this is called duplexity theory, where we've got two different kinds of receptors. The theory is that they're going to mediate different types of vision. There's special terms for this: photopic and scotopic. This is like a real photograph, and then scotopic is less sophisticated than a photograph. But I just need you to know that there's different vision mediated by cones and rods. Here's kind of a list of what's different about the two, and you can pause the video, kind of study this if you'd like to. But you can see that cones work best under high light condition. You turn the lights on, whereas rods work better in the dark. Cones see color and rods don't. Cones are clear, high acuity. Rods are kind of blurry. Cones are low sensitivity, right? We can't see in the dark with cones, okay? Whereas rods are high sensitivity, we can see in the dark in cones, okay? Cones are concentrated in the fovea in the center of your eye, whereas the rods are concentrated in the periphery to the outside. So this is why you can see clearly and see in color straightforward, whereas things in the periphery are more blurry. You might not be able to see the details or the specific color gradients and that kind of thing off in your peripheral vision. And the last feature is going to be maybe the most important, and that's that they differ in convergence. Talk about that on the next slide. So the reason why cones and rods work differently is because of a phenomenon called convergence, or because of differences in convergence. For cones, which show up in the center of your eye and allow us to see clearly, each cone pretty much gets one retinal ganglion cell, one neuron to one cone. Whereas it'll be a number of rods are going to converge or come together onto one neuron. And there's a couple consequences of this, right? One is that the cones are more specific. So every signal from this retinal ganglion cell we know came from this one cone, which means the light came from this one place in the world, right? So one cone, one neuron, one ganglion cell means that the signal is very specific. I know that it's coming from, this part of the letter L, right? Rods are less specific, so it's going to be less clear. I'm not going to be quite sure whether the light's coming from maybe this part of the letter L, or maybe it's coming from down here or over here, right? Because various different rods could be responsible for the signal coming to that neuron. So it's going to be less clear, it's going to be more spread. But on the flip side, sensitivity is better with the rods. So if I've got in dim light and only a little bit of light is coming back, I might have 5 rods telling me that there's light, whereas I would only have one cone being able to respond. Okay, so 5 voices are louder than one voice, right? And so I have good sensitivity with rods, which is why when you see in the dark, you can't see in color and you can't see very clearly. Your rods are activated to allow us to see it all. So the rods and the cones are going to take the signal. They're going to send it down the bipolar cells to the ganglion cells, which are then going to be the first neurons that are going to run down the optic nerve. The next steps are 3. Well, 2 more. 2 more steps. So the first step is we go from the retina, right? The next step is to get to the thalamus. So we talked about in chapter 3, the thalamus is a sensory relay. And here we're doing it now, we're relaying. So the thalamus is sitting in the center of your brain, and there's a specific nucleus. The lateral to the outside geniculate nucleus is located in the thalamus. So the signals from the optic nerve is going to go through that thalamus, and it's going to eventually get to the occipital lobe, to a very specific part of the occipital lobe called the striate cortex. Striate means striped. There's like a striped area of your occipital lobe. That's where it goes. So retina, geniculate, striate system. And there's an interesting thing where signals from both eyes are either going to cross and head to the opposite hemisphere or stay on the same side to the same hemisphere. Okay? And the pattern that it works is a little bit complicated, but generally one way to think about it is that light that's coming toward your nose is going to cross and end up in the opposite hemisphere. Okay? And then light that's going to go laterally to the temples, to the temporal sides, right, is going to stay on the same side. Here's a figure depicting that. The consequence of this system, right, is that the hemispheres are going to get signals from the opposite visual field, so the opposite parts of the world. So signals from over here in my right visual field are going to make it to my left hemisphere. Signals from over here are going to make it to the right hemisphere. It is not the case that signals in my right eye always go to the left hemisphere, or that signals from my left eye always go to the right hemisphere. Okay? That's not true. Okay? The right eye is going to send sub-signals to the right hemisphere and sub-signals to the left hemisphere. The way we accomplish that is that the nasal hemiretinas, the middle parts of the eye, are going to cross at the optic chiasm. Okay? So things off into the right are going to hit my right eye toward the middle and cross. Things off into the left are going to hit my left eye in the middle and cross. Okay? In contrast, signals that hit the outside, the lateral parts of the retina, are going to stay on the same side. So stuff in the left visual field hit the right part of my eye and stay on the right. Things from the right visual field hit the left part of my eye and stay on the left. Okay? So when you think about this, the left hemisphere is always going to get signals from the right visual field. It's going to get it from both the right eye, hitting in the middle, and the left eye hitting on the outside. The right hemisphere is going to get signals from the left visual field, both from the right eye hitting on the outside and the left eye hitting on the inside. Finally, the signal is going to make it back to the occipital lobe, right and left hemispheres. There's an interesting additional phenomenon here where not only are we getting signals from the right and left visual fields, but we're also getting signals from the center of our visual field, right? The top, the bottom, the corners, right, of our visual field. And our occipital lobe is laid out in this retinotopic layout. So depending on where the signal hits my retina, there's going to be a designated spot of the cerebral cortex, of the occipital lobe here. Interesting other phenomenon is something called cortical magnification, where the area of the cortex dedicated to the center, or the fovea, is larger than you would expect. So if we look at this over here, signals in the center at number 1, right, are going to end up in the right hemisphere. They're going to go upside down, so they're going to end up over here. You see how much of the brain is dedicated to this tiny little area 1. Then we compare that to this whole area 9, all of this retina, right? And you can see 9 gets this amount of area in your brain. So the same amount of brain is dedicated to this tiny area as is dedicated to this whole area 9. So we're essentially zooming in. What we're actually getting in our brain is essentially like a zoomed in picture of the center and then a zoomed out picture of the outside. We're biasing our brain processing toward the fovea itself, where all those cones are, where we can see in color, and where they're converging on neurons. Okay, so we've made it. We went from the retinas back to the thalamus, the geniculate nucleus of the thalamus. We crossed the optic nerve when the signal comes to the center of the eyes. And then we made it back to the occipital lobe, specifically the striate cortex. And there we have a map of the world, or a map of our retinas, that's blown up in the middle with cortical magnification.